Hi everyone, good morning, good afternoon and good evening depending on where you are joining us from. My name is Richard Thompson, a scientific editor at Cell Report Sustainability. Together with my colleagues Michelle Muzio, the editor-in-chief at Chem Circularity, and Shanshan Zhang, a scientific editor at One Earth, we welcome you to the Cell Press webinar series, Forums on Sustainability. Today's webinar is themed on securing a resilient future for agriculture through connections between technology and practice, where we'll be delving into the urgent challenges and opportunities facing global agriculture in a changing climate. As extreme weather events strain water supplies and food systems, it's clear that new solutions are needed. Solutions that draw on nanotechnology, chemistry, engineering, and broader circular innovations and governance. Today, you'll hear from leading experts who will share the latest advances in fertilizer and pesticide technologies, soil health, toxicology, and much more. Our panel will discuss how integrating innovative advances in science and policy can drive sustainable, resilient farming practices and safeguard food security worldwide. We will first invite our keynote speaker to set the scene for the forum before inviting each of our speakers to share their latest research. We will then dive into a live discussion across the panel. So just briefly, I'd like to highlight Cell Press's portfolio of sustainability-related journals. So besides the journals that my colleagues and I represent, being One Earth, Cell Report Sustainability, and our newest community-focused journal, Chem Circularity, several other journals frequently publish sustainability-relevant content. All of our editors consistently communicate with each other, and we cross-promote work published at Cell Press. So please do feel free to be in touch with an editor about any of the journals across the Cell Press portfolio. Now, before I hand over to our keynote speaker to open the forum, I'd just like to mention some logistical details for our audience. If you have a question for any of the speakers, please use the Q&A box on the right of your screen. You can enter a question at any time, or you can upvote questions from others that you'd like to be answered by clicking on the thumbs up just under the question. We will try our best to pass on as many of your questions to the speakers as we can during the Q&A. It is helpful if you also include the name of the speaker to whom you would like to direct your question. And last but not least, the webinar will be recorded and it will be available on demand on the Cell Press Sustainability Hub after the event. So without further ado, I'm delighted to now introduce our keynote speaker, Dr. Jason White. So Dr. White is the director of the Connecticut Agricultural Experiment Station. That's the oldest agricultural experiment station in the United States. There he leads a team of around 105 scientific staff and manages an agency budget of around $13 million. In addition to this role, Dr. White also holds appointments at the Harvard University T.H. Chan School of Public Health, and he is a commissioned official of the United States Food and Drug Administration. His expertise are in applying nanotechnology to agriculture, especially using nanoscale micronutrients and materials to help suppress crop diseases, boost tolerance to environmental stresses, and increase food production. He also researches how nanomaterials accumulate in crops and their environmental fate and toxicity. And with that, I'll hand things over to you, Jason.
Thank you. I just tried to share my presentation and didn't open, so let's try that again. It says it's trying to open.
There we go.
There we go. Bingo. Okay, well, thank you. I'm very excited to be here today to talk about what I think is a really critical issue, but also I think this multidisciplinary approach that we're taking to discuss the issue in this virtual webinar is really a great way to do this. So I'm going to talk about some of the work that we've been doing over the last 10 years or so, looking at how nanotechnology can increase food production, but also make the system more efficient and more circular. Let's get started. So I think the best place to start is to kind of set the stage. And this is a paper from a couple of papers we published. The first one was back in 2019, where we kind of set the current status of agriculture. And we know from the last 70 years, the Green Revolution obviously significantly increased agricultural output, food production. Green Revolution being the development of irrigation, pesticides, and fertilizers. But we know this gave us a system of conventional agriculture that's completely non-circular and highly inefficient. And these are just some examples that we highlighted in this paper, one being 70% of all fresh water use every day goes to agriculture. We know that the rate of crop yield increase has been on decline for decades. That's this data you see over here where we looked at 11 crops. We know we have significant problems globally with both hunger and micronutrient deficiencies. Yield potential, which is the amount of food, for example, a piece of land could produce. The best we do is 80%. Often it's quite a bit lower. And then the piece we actually started working on about a decade ago was this inefficiency of agrochemical delivery. Most fertilizers, you're looking at about 40% or so efficiency, some pesticides down to 1%. And we got to thinking about nanotechnology because a decade ago in nanomedicine, what was being done was nanotechnology was used to precisely deliver cargo in a complex system. And in a lot of ways, our problem in agriculture is somewhat similar. So that's what got us going on this. But here's the real motivation, and we've known this for over a decade, that we're going to need to increase food production by up to 100%, depending on whose number you use, by 2050 to feed the population we'll have. And we're going to have to do it really in the face of significant pressure from a changing climate and other factors. And in fact, climate change is actually making this more difficult more quickly than we thought. There's no species that's better at getting in its own way than humans are. So obviously we have the human conflicts that make this harder. So many of us in this space, and I think you'll hear this today, have really come to the conclusion that we need to completely transform how we grow, distribute, and store food on the planet. And that nano-enabled strategies can really be a big player here. This is actually not a full list of the approaches that can be used. This was just what I could fit on the slide. I'll show quick examples of a lot of these from our work and from others, but some things I'm not going to touch at all today that are significant, like minimizing contaminant accumulation, sensors, extending shelf life. So I think there's a range of possibilities here. This last thing I'll mention on this slide, this is from a paper that we published in Nature Nanotechnology back in 2022. And why I mention it is most of the authors actually worked for the US EPA, where we specifically looked at nanopesticides and just kind of the baseline technology. You were getting anywhere from a 20 to 30% increase in efficacy, but also reducing loss of the active ingredient and reducing damage to non-target species. So I think that kind of sets the stage of what could be possible here. So at the experiment station, we've got a pretty large program in this area using nanotechnology in agriculture and food. My PhD is actually in in environmental toxicology. So when I started hearing about nanomaterials in the food supply, I thought that was a terrible idea. So I did what any toxicologist would do, which is I set up a nanotox research program. And we published our first paper in 2009. And we still have research in this space, but what we noticed after a couple of years was that not only were there certain instances where nanoscale materials were not toxic to crops, to plants, but there were actually instances where we could see significant benefit. It obviously depended on the material you were using, the chemistry of that material, the crop, the exposure regime, and all of that. But that really gave rise to this other area of nano-enabled agriculture, which has become a much larger research topic for us. Now, what's interesting as a toxicologist is that, for a given material, the difference between causing toxicity and causing benefit is actually really narrow, and it's mediated through reactive oxygen species homeostasis a lot of times, ROS. So that's kind of where we started focusing a lot of our work. So this was from a paper that we published in 2022 in Nature Food, where we kind of looked at the work we've been accumulating over the last 7 or 8 years and kind of reframed it in terms of how are we going to increase efficiency, yields, and enable circularity in agricultural systems. And that's where this concept of controlling the ROS reactome really came about for us. Now, reactive oxygen species, they're generated during normal cellular metabolism, hydroxide, hydroxyl radicals, so forth and so on. But they're also generated when a plant or any organism experiences a stress. So the ROS will damage biomolecules, organelles, so forth and so on. I think we all know this. But what's important is that lower concentrations, these ROS are actually important signals, intracellular signals. And that's where we really got this concept of the reactome. Could we control the ROS homeostasis in plants to get a benefit that we needed? So we approach this two ways. One is a scavenging way. So a plant experiences a stress. Can you add a nanoscale micronutrient? That's what we ended up using to alleviate that stress, either directly from the material itself scavenging the ROS or from a stimulation of pathways that'll take care of that. So it's a recovery pathway. And I'm going to show you a lot of data. We're going to go through it quickly, and the specifics don't really matter. It's more the platform that's important. But then the other approach, which I think is even more exciting and we have far less data on, is this preventative strategy. So can you add a small amount of something that triggers all of these stress-related pathways and then prepares that plant or that crop for a stress that's going to occur later? So again, this is controlling the homeostasis around oxidative stress. And really what we're trying to do is build climate resilience into these crops. So we first started working on this back in 2014, and we decided to work on soil-borne fungal diseases because they're a big problem in the U.S., very difficult to control. In the U.S., we're spending $600 million a year on fungicides, and we're still losing $200 million worth of food. So we wanted to really focus on these nanoscale micronutrients because we know that the micronutrients themselves are important parts of plant defense and plant immunity, but it's really difficult to get these micronutrients in the plant at the right location, at the right time, and the right dose, which is really where nanotechnology is going to come in. So we like to highlight that although we're working in disease systems, we're not really targeting the pathogen directly. We're targeting the ability of the plant to defend itself against a pathogen. So this was the first paper we had in this space. Some very simple experiments to begin with. We published this in 2016. We focused on eggplant and tomato because those were two systems we had good fungal systems with. We do very simple foliar applications of these micronutrients. You always have to use the conventional controls. You should not be using a nanomaterial unless the nanomaterial is better, unless it gives you something unique. So a simple foliar application to a seedling, and we're an ag experiment station. So we've got 3 farms. We can do lots of field studies. So the green over here is copper. The center green is the nanoscale version. And what we saw was that, that foliar treatment of the seedling, we got increased yield, we got greater disease suppression, and we got higher content of copper in the roots, which is where we needed it. And we did that at a dose that was not even toxic to the pathogen in vitro study. So it's all around this ROS. And this ability of the plant to defend itself. And again, it's a field study, so we can do these calculations. We spent about $44 US per acre for nanoscale copper. And in so doing, we got about a $10,000 per acre increase in yield, increased food production. And what did we really do? We took a nutrient that the plant needs and we just gave it to it in a different form. So that really kind of set us off to the races. This was from a paper in 2020 in Nature Nano where we really kind of nailed down that mechanism. We were part of an NSF CCI chemistry center that actually just ended after 13 years and ended September 1st. But in the center, we really started focusing on the chemistry of our materials. So this again is copper. We switched to soybean and soybean sudden death syndrome, which is another fungal pathogen. In these greenhouse studies and in the field, this pathogen will reduce yield and photosynthesis by up to 70%. But again, our foliar applications, specifically of the nanoscale copper, were alleviating much of this damage. This was our first study where we really got into gene expression. So we're doing a foliar application of the copper. We're measuring a transcriptional response in the roots. And in this case, it's almost 24 different pathways or genes that are upregulated to give this plant better ability to defend itself. And what was really interesting is that not only was the nanoscale size a critical part of this, but actually the properties of your nanomaterial. You could control shape, you could control dissolution profile, and you got different levels of activity, which really gets at this concept of tunability of your application. So some of the other materials we've been working a lot with under biotic stress, disease stress, we have a lot of work on sulfur through some USDA grants. So at this point, a lot of these experiments are going to look similar, and the specific data isn't that important. I'm just going to give you a kind of a high level overview. Again, you always have to use your conventional controls. We do greenhouse studies so that we can get at mechanisms. So we'll do orthogonal endpoints for 20 or 30 or 40-day studies measuring not just physiological parameters, but also time-dependent metabolomics, time-dependent transcriptomics or proteomics, things like that. And in this study, that ended up being really important. So I mean, this is tomato and fusarium again. We're getting an 85% 90% reduction in biomass, and we're able to alleviate much of that with these nanoscale amendments. But importantly, by using our two-photon microscopy plus these time-dependent omic endpoints and ICP, we're actually able to show that the typical sulfate uptake pathway for plants was not being used. Because it was a nanoscale elemental form, we were actually feeding the sulfur directly into the glutathione pathway, which is a defense pathway. It's much more efficient, and it really led to the plant being able to better defend itself. And again, here's your math that supports our study. You know, we spent $33 on nanoscale sulfur per acre, and you know, we're going from 4,900 kilograms of tomatoes to almost 12,000 per acre in a healthy system. And there's the disease data. So the data to a certain extent speaks for itself. So you can't talk about the inefficiency and non-circularity of agriculture without talking about nitrogen and phosphorus, and we are working on those. Nitrogen first, you know, we spend a huge amount of resources to generate nitrogen fertilizers by the Haber process. Our focus is looking at biological nitrogen fixation, where you've got the small number of microbial species that can do this process for you. They can take dinitrogen gas and make ammonia. There's a couple of issues with that. One, BNF is inherently repressed by all of the synthetic nitrogen that's being added. Plus, BNF is very sensitive to environmental stress, which gets us to climate issues. So that's really where we're focusing. We're looking a lot at things like molybdenum and iron, because those are micronutrients that are key to the enzymes in the nitrogen fixation pathway. This is just a couple of the studies, and this afternoon we're actually going to be submitting a USDA grant on this topic. But you know, we see things like at the early stages of soybean growth, you know, your nanoscale molybdenum disulfide can act directly as a nanozyme. It can quench the RLS from any sort of stress that's happening. But later in the life cycle, that slow controlled release of molybdenum beyond the conventional fertilizer really gives you prolonged activity of your nitrogen fixing bacteria in your nodules. So you get delayed senescence, you get greater activity. With phosphorus, this is the perfect example of non-circularity. I mean, there is a finite amount of phosphorus to be mined on this planet. Depending on whose number you use, it's 100 to 300 year supply left. So, you know, 80% in most cases of the phosphorus that's put on a field is not going where it's needed. So our strategy here is to take all of the phosphorus that a plant or a crop needs, and instead of just putting it in the field, put it in these nanoscale biopolymers. And you know, we've used everything from Zane to chitosan to nanoscale cellulose. You can really control the chemistry of these polymers, which gives you an ability to control the when and the where of phosphorus release, which is in a lot of ways crop specific. You can use particles, you can use fibers, you can do gas phase esterification to put hydrophobic coatings onto your carriers to again tune that release. And we can see things like up to 90% reduction in phosphorus loss from soil where plants are growing. So we've got 4 different projects actually with a whole range of collaborators looking at different phosphorus strategies. So other micronutrients that we're looking at in terms of biotic stress, I mentioned iron, but we're looking at selenium, magnesium, and lanthanum. But one interesting thing we're really getting into now is this green biosynthesis. So you can synthesize nanoparticles of micronutrients in a number of ways, one of which is in the presence of plant or microbial extracts, which give you the reducing power. And why do I have circularity highlighted here? We're actually starting to use agricultural plant waste streams as source material. Now, if you're making micronutrients, you're not going to use that much. But if you can develop technologies to make nitrogen and phosphorus-based fertilizers out of plant waste streams, then I think you're really onto something. So this is a space that we're going to start spending a lot more time in. So this also works with abiotic stress. So the previous work was disease and pathogens. This is more climate-related stress. I'm going to go through these really quickly because I know I'm running out of time. But drought is obviously a big one. Again, different forms of nanoscale copper, including copper sulfide. So you're getting two nutrients for the price of one. But this data is starting to look very similar at this point. You get a stress. In this case, it's a drought stress. It's decreasing significantly just about every endpoint you want to measure, in this case in soybean. But again, supplying these materials at the right dose at the right time with the right chemistry, you can alleviate much of damage, and it's all around these ROS related pathways. So that is copper. You can also do this with silica. This is a paper that just came out, actually. And this was interesting because we started, again, really focusing on the chemistry of your nanoscale silica. And it turns out the porous and the hollow materials end up doing much better in terms of release profile, in terms of alleviating drought damage. So we're actually going back and we're taking a look at these porous and hollow structures and now using them as the skeleton, as the backbone. So we can start decorating them with other micronutrients, things like zinc or boron or other things, to kind of give you this multifunctional benefit. Another stress we looked at was salinity. This is actually from a paper in 2021. We've got one we just submitted on lettuce, but this was cotton, because we got money from the cotton board, so you have to use cotton. But the strategy was a little bit different here. So instead of doing foliar applications, now we've started doing seed treatments, which is really ideal if you want to deal with the agricultural community. Because, you know, if you're going to ask a farmer to do something additional like a foliar application, you know, that's more work and more money for them. But if you can treat a seed and they're just buying a different seed, then that makes this a lower barrier to get over. But the approach is exactly the same. You know, in this case, we use cerium, which is not a nutrient, but it's a known antioxidant, and you get decreased ROS in these seedlings when they're exposed to your cerium oxide nanoparticles, and you see better growth. Another stress that we're looking at is light or UV stress in certain areas of the world. This is a big problem. This is a project I have with Cutter, where we're looking at a couple of different things. You can do the same type of approach. You can use nanoscale micronutrients. We've got this multi-micronutrient composite as a foliar application to stimulate pathways and prevent ROS-related damage to the organelle, to photosynthesis. But you can also use other materials like carbon dots, which you can decorate with other micronutrients that will actually do this photoconversion of light. They'll take ultraviolet light wavelengths and convert them to visible light. So you're doing two things. One, you're preventing damage, but two, you're feeding more electrons into the photosynthetic system. So that's actually a pretty exciting strategy. So really quickly, I'll just talk about the other approach. So that was all recovery. Stress first, then alleviate the damage. What about this other instance where can you prepare a plant either by seed priming, leaf priming, or both? Can you stimulate these pathways first so that a stress that occurs later, the plant's better able to handle it? I would have said no initially, but the answer is yes. This is our first paper we published with rice, where we looked at a fungus and a cold stress. So we do a seed priming or leaf priming. My collaborators on this are in China, and they really like silver initially. We've moved away from silver now, but use a small amount of silver in these training or these kind of pre-treatments. You stimulate the ROS related pathways, and this is the disease damage. If you can see over here, this is rice blast in the control. So this is the seed, the leaf, or the seed leaf priming together. So this pathogen shows up about a month after you do this priming, and you're able to reduce disease damage by over 50%, and you're increasing cold stress tolerance. Again, just by training or priming these plants for a stress that's going to occur later. And if you start thinking about, you know, climate resilience, that's really a pretty exciting strategy. This was a more recent paper where we looked at maize. We switched to iron. We started doing single and multiple stress conditions, but the result was still the same. You could still get this better ability of the plant to defend itself against disease. This is our most recent paper where we looked at silica. So I think I'll just skip this because I know I'm short on time. So, you know, this is some really cool science, but can we actually get it into the field? We started publishing a lot in 2017, 2019, got the attention of a number of agrochemical companies. There was a lot of interest, but then nothing. And then COVID happened. But towards the middle of 2023, you know, these companies started reaching out to us again. Now we have active funding from a number of these. We've actually formed a spinoff company called Convergent Bioscience. You're probably asking yourself now why I didn't do the disclosure, because it's a company that has no employees or money yet. So that should change within a month. So this is probably the last presentation I'll do without a disclosure. But the point being, you know, what's changed here over these last few years? And I think some of it is that the science has matured. I mean, this is the number of publications in this space. But the other thing that's happened, I think, is just a general recognition of the magnitude of the problem. I mean, that light at the end of the tunnel, that's a train, and it's coming right at us. So figuring out how we're going to feed the world in a changing climate, both sustainably and with circular systems, is really going to be one of the most significant challenges we're ever going to face. So I will quickly conclude. So hopefully I've set the stage for what I think are going to be some really exciting talks on this topic, and how we can really kind of redefine and transform how we are growing and distributing food on this planet, and being able to do it in a way that's resilient to the negative impacts of climate is going to be critical. I have a large number of collaborators and acknowledgements. I'm just a spokesperson in a lot of ways. So I will stop sharing my screen now.
Thank you, Jason, for starting the forum with such an excellent talk and really setting the scene for this forum and setting the context of the challenge we face. That was really fascinating. I'd just like to encourage all of our audience members to add their questions to the Q&A box, and we'll get to them shortly in our panel discussion. I'll now hand over to my colleague, Michelle Muzzier, to introduce our next speakers.
Yeah, no, thank you, Richard. Thank you, Jason. That was amazing. I am honored to announce our next speaker, Professor Iseult Lynch from the University of Birmingham, an environmental chemist working at the interface of chemical and materials pollution and environmental policy, adding a lot to our discussion today from the angles of toxicity and safe and sustainable by design. Professor Lynch is the director for research at the University of Birmingham Center for Environmental Research and Justice, an affiliate of the Birmingham Institute of Sustainability and Climate Action. Take it away, Professor Lynch.
Thank you very much, Michelle and Richard. It's a really great pleasure to be here. And it's a challenge to follow Jason, of course. And the eagle-eyed among you may spot a figure that I have on a full slide that was in the top right corner of one of Jason's slides, because of course we have worked together over a number of years. So I'm going to talk a little bit about our work on leveraging nanomaterials safely and sustainably, or circularly, which is a hard word to say, for soil health and for plant health. So we've already touched on this from Jason as well, but in reality, food production is responsible for 1/4 of the world's greenhouse gas emissions. And as Jason has already indicated, as we scale up the production of food to feed our growing population, that's only going to get worse unless we actively engage in ways to produce food more efficiently and in a way that produces less greenhouse gases. So this was from a paper not that long ago in Nature that gave that data, and also some of the numbers come from the website Our World in Data. Much of this is driven by the overuse of fertilization and fertilizer. So one of the challenges we have is that as we add artificial nitrogen into the system, so since the discovery of the Haber process, we are actually driving the system and making those plants that are able to pull nitrogen from the atmosphere and fix it into the soil, we're basically making them lazy by giving them the circumstances where they don't need to do that, because there's now excess nitrogen in the soil because we're adding it continuously. And the other main challenges are, I think Jason already alluded to it, but the amount of the nitrogen that actually gets taken up by the plants versus the amount that gets lost into the atmosphere as greenhouse gas, or that runs off and causes water pollution, runs off into local streams and things, is pretty high. So we're producing all of this nitrogen at great energetic cost and putting it into the soil, which is causing some of our plants and their symbiotic microbes to become lazy. And therefore, then we have to just keep reiterating that cycle. So we really need to find ways to move away from that. And one of the ways we can do that is, as Jason mentioned, through targeting our delivery of nitrogen. So rather than just having it abundant in the soil, really target how we get there. So over the last few years, we've also been thinking about how we make agriculture more sustainable and how we enhance the circularity of food production. So this is the figure that you will have seen in the top right-hand corner of one of Jason's slides, because it is a collaborative work. But I just want to spend a couple of minutes, well, a minute maybe, talking about some of the strategies here. So this is work that was led from my team by Dr. Peng Zhang. And some of the things we're doing is really using the nano delivery as an alternative to gene targeting, or to gene editing, or things like that. So nanotechnology is an alternative to some of the concerns potentially about genetically modified plant species. So by being able to target the nutrient or the fertilizer or whatever we're delivering to the desired location, that's a potentially much more acceptable solution. But of course, the challenge then we have to think about is, how do we make sure that there isn't residues of these particles in the edible parts of plants? So that's also something that we're thinking about, how we can design those nanomaterials, either using the strategy that Jason mentioned of using elements that are already essential elements for the plants, or using ones that will degrade or transform back to inert or non-toxic components. We're also using these nanomaterials then to stimulate regulation of the biological nitrogen fixation, BNF process. And we can have sort of strategies where we target directly the foliar application, or into the soil rhizosphere, where we're interacting directly with the symbiotic microbes related to the plant root system. Some of the materials that we use are also to have the ability to interact with light, so they can trap the sunlight and use that for some of the energy conversion reactions, so to enhance photosynthesis and processes like that are also involved in biological nitrogen fixation, and then also really just enhancing the availability of some of these macronutrients as well. So that was published a couple of years ago. And again, was using these molybdenum sulfide nanomaterials, so using both to enhance the yield of the crop, but also then the nutritional quality, because it isn't just about enhancing the amount of the plant, or if the plant is taking up more water and is therefore by mass heavier, but actually not an efficient source of energy, then it fails as a food substance essentially. So soybean, of course, is a crop with global significance. It's a staple in very many countries. It has a low reliance on nitrogen fertilizers because it has the capability to perform biological nitrogen fixation. But that capability to undertake biological nitrogen fixation is compromised by abiotic stresses. So part of the work that we've been doing is looking at how, if we have a nanomaterial form of the molybdenum sulfate versus each of the molybdenate or the sulfate individually, how in the vegetative state we can enhance the plants and the root system's ability to scavenge these reactive oxygen species, and then also to enhance both the photosynthesis and the nitrogen fixation and nitrogen assimilation. So really performing some molecular and mechanistic level studies to really understand what's happening and how those systems work. But also, I don't show the data here, but we also then do look at the calorific intake, or the calorific values of the resulting plants. Another aspect we've been taking, so several of the projects that I've been working on over the last while have really focused on the idea that we're generating large amounts of data, and the informatics tools and the use of artificial intelligence can really help us then to identify new ways of new patterns in the data, but also then how can we use that information to design our materials more effectively. So in the same way that we've had precision medicine emerge as a field, there's now also the idea of precision agriculture. And that's precision in terms of the delivery, but also precision in terms of tailoring solutions for specific soil types for specific climatic conditions, specific, temperatures or rainfalls, and so on. At the moment, of course, that data is all hugely disparate and all over the place. So, we have some work around organizing the data for nanomaterials properties. There are some databases perhaps on, some of the soil parameters, the soil chemistry, some growing amounts of information on how the plants respond. So, for example, what the roots secrete out under normal conditions and under stressed conditions, and similarly the microbial secretions. There's also a growing body of data on uptake, both of the nutrients and of different nanomaterials, and the translocation, transformation, and trophic transfer of those materials. And then, the body of data we have on various different types of plants and so on. So our idea was that if we can develop systems that allow us to integrate all of that data and interrogate all of that data, we can then use it to answer all sorts of questions and optimize the design of our experiments. So within the NanoCommons project, and the NanoSolve project, one of which was NanoCommons was really around the database, and NanoSolve was more around the tools. We sort of envisaged this idea that if we could develop modules that, a first module would extract data from existing databases and organize it in a way that it could be reused, a module for calculating descriptors around the nanomaterials, a module that would look at, that would automate the modeling itself, depending on what it is we want to model or how we want to further enrich the data using, for example, QSARs or first principles models. And then that would also give the domain of applicability of the model so that it could be reused. And with this vision, then we could think about, using that to design optimized nanomaterials to, for example, enhance crop yield, or to maximize the nitrogen use efficiency, or to improve the photosynthetic efficiency, or so forth. So this is a sort of a vision, and I think it links nicely also to some of the growing capability of using nanomaterials themselves as sensors for plant health. So in due course, we could kind of think about, instead of applying fertilizer as a precautionary measure, that you would have sensors that would tell you based on what your plant is exuding, when you need to add fertilizer. So a much more precision generated approach. And then just the very last things we've been working on, this was published in Matter just earlier this year, is linking then to the Safe and Sustainable by Design framework. So this is an approach that thinks about the whole life cycle of a material. So what's involved, from the extraction phase to produce the material, then as the material gets used in specific products, and then end of life, and how we can potentially recover some of those materials and reuse them again. But also then, as Jason already alluded to, can we produce the materials in the first instance using either crop waste or, bioavailable materials, biorenewable materials. So linking to the green revolution, and understanding then if the materials that we make from biorenewable sources are safer than those we might be making from resource limited or critical raw materials, for example. So that's thinking about the material production, but then also during the use phase as well. Can we understand, for example, the transformations that those materials will undergo and design those transformations in? So the more we understand about how the materials are taken up and translocated, the more information then we have to design, okay, what enzymes might be there, what pH might be there, what salinity might be there. So can we tailor materials to biodegrade in specific places, taking account of the transformations that they naturally undergo? And one of the systems we're looking at the moment are these metal organic frameworks, which were initially largely designed for use in the gas phase, but now we're increasingly looking at materials that can be stabilized in aquatic systems, and therefore also in soil systems. So hopefully, building on what Jason has already shown, I hope that I've shown you that nanomaterials do offer some really promising technology developments. I didn't actually talk about the energy security here, but a lot of what we're doing also is then linking through to things like solar desalination. Can we capture those runoffs from farms and remove or concentrate out the potential pollutants so that water can then be reused or recycled? We're also doing some work on electrolysis of the brine that's produced and things like that, but also that consideration of the safety and sustainability profile of the materials from the outset, and designing those materials to be as benign as possible, to transform in ways that we either control or predict, and to be circular is essential. And that nanomaterials offer numerous ways to enhance the performance and yield, but also then that fair data and nanoinformatics and AI will play an increasingly important role in all of this. And with that, I will, of course, just thank the funders and again, just acknowledge Peng and Swaroop, who are the hands in the lab. And thank you.
Thank you so much. That was a wonderful overview, and I really love the point at the end about design, and I think we'll come back to that in the discussion. Thank you so much. Our next speaker will be Professor Kimberly Parker from Washington University in St. Louis. Professor Parker's research group is advancing engineering and chemistry to address emerging environmental challenges. Current research in Professor Parker's group continues this multidisciplinary aspects of this webinar today, bringing together environmental chemistry and engineering to enable the safe and sustainable use of next generation biotechnology products. So take it away, Professor Parker. Thank you.
Fantastic. Thank you again for this invitation and for the audience online. I'm really excited to engage with you all. Yes, I'm going to switch gears today to talk about a different type of technology that's currently widely used in agriculture and I think is an important part of the future of agriculture, and that is agricultural biotechnology, things like genetically modified organisms or crops that are a really important part of our current agricultural system. So when I started my research group at Washington University in St. Louis in 2018, I really wanted to focus on environmental and agricultural challenges that are relevant to our local region. And in my area, my part of the world, we have really big, large-scale agriculture for things like corn, soybean, and cotton. And agricultural biotechnology or genetic engineering is a huge part of how those crops are currently produced at scale. These data are from the U.S. Department of Agriculture, and they show the percentage of different crop species that are planted as one or more genetically engineered variety across the U.S. You can see here that if we look over the last decade or so, really we've been holding stable at around 85 to 95% of crops like soy, corn, or cotton being planted as a genetically engineered variety. And so this is a really critical part of how agriculture is currently practiced in the US, and like I mentioned, how I think it's going to be practiced going forward. And so as an environmental chemist, I want to move beyond the discussion of is GE good or bad, and really think about what are the specific environmental challenges and solutions posed by these crops going forward. And today I want to share some of our work specifically on the crops I've highlighted here that are all labeled HT. HT stands for herbicide tolerance. And so we can see HT in soybean, cotton, and corn, all at really high market penetration rates across the US. So what is herbicide tolerance and why is it so popular among farmers? Herbicide tolerance allows farmers to control weeds in their field using chemical herbicides without damaging their crop. You basically genetically engineer your crop so it can withstand that herbicide so you can kill all the weeds, but your plant still grows perfectly healthy. The first of these was developed for the herbicide glyphosate in 1996. You may be familiar with its trade name Roundup, and this application option is really one of the major reasons why glyphosate use has really shot up relative to other herbicides, and it's currently the most popular herbicide used both in the US and around the world. The challenge is that our agricultural systems have become increasingly reliant on glyphosate use on glyphosate-tolerant crops, again, due to this widespread popularity and importance in efficient, large-scale agriculture. At the same time, those very same tools are becoming less and less effective. we've seen glyphosate-resistant weeds crop up across the US and around the world, meaning we can no longer use glyphosate to control those important weeds pests. And this is something I like to call the herbicide resistance cycle. You may be familiar with this concept from antibiotic-resistant bacteria, for example. We use these chemical tools to control pests. That selective pressure allows those pests to evolve resistance, and then we need to go out and find new tools, which then repeats the cycle again and again. And so we see the same trend happening in agricultural pests, including insect pests, and what I'm focusing on here, these weed species. This is an existential threat in agriculture. We cannot continue to produce food at scale at the way we rely on agriculture to currently function unless we can solve this herbicide resistance cycle, or as we currently are doing, developing new tools that can continue to control those weeds. And so over the last decade or so, there's been a huge effort in the agricultural industry to develop new GE crops with new herbicide tolerance traits to allow new herbicides to be used in those same ways. So over the last few years, we've seen herbicide tolerance traits getting commercialized for herbicides like 2,4-D, dicamba, isoxafludol, and there's even a few more in the pipeline that will start to be planted starting next year in the 2026 growing season. And so I want to focus today on one of these, dicamba, and some of our research to really address some of the key new environmental impacts that have emerged along with these dicamba tolerance traits. So the dicamba active agent herbicide was actually first registered in the US in 1965, so we've had it for a while. The new thing is that in 2015, these herbicide tolerance traits came on the market. And to give you a sense of how popular they were in the US, 2 years after you could first buy them, already 1/3 of all soybean and over half of all cotton in the US was planted to express dicamba tolerance. And we see this hockey puck effect in terms of dicamba use, where we rapidly accelerated the amount of dicamba being applied on major crops like cotton and soybean. Unfortunately, over that same time period, we began to see these environmental impacts emerging from this widespread dicamba use. These were called, this was attributed to the phenomena of dicamba drift. Essentially, you apply dicamba over here on your tolerant crop, but it picks up and moves through the atmosphere to deposit on non-tolerant crops or other sensitive vegetation. And so we saw these news stories reporting damage across the US in agriculture year after year due to this problem of dicamba drift. One report that I've been thinking about a lot lately came out of the Illinois Department of Natural Resources. They looked at trees systematically across the state of Illinois, where each color circle on that map shows a different site that they visited. At over half of the sites, they found evidence of herbicide damage, and what they classified as severe damage, at 99% of the sites they visited, they found evidence of some herbicide damage. And what they classified as severe damage at around 54% of the sites. And so we see these ecological impacts resulting from dicamba drift as well that we're still just starting to understand. And so what am I doing as an environmental chemist? Well, my group is trying to fight this issue of dicamba drift. One of the key things we've been working on is trying to understand what are the fundamental processes that are guiding dicamba drift and causing this widespread damage in crops, trees, and other significant vegetation. We also put on our engineering hats to try to apply environmental chemistry to solve the problem. How can we design more effective chemical formulations that hold dicamba where you want it, preventing it from going into the atmosphere and causing this off-target damage? Lastly, we're trying to set our sights on the future. We know more and more of these herbicide tolerance traits are going to be marketed and used as we continue that herbicide resistance cycle. What will their new environmental impacts be, and what can we learn from our experience with dicamba to manage and prevent those impacts to health and the environment going forward? One of these we've been working on a lot recently is the herbicide isoxafludol. This is an important carcinogen and reproductive toxin that's starting to show up in our water, and we expect to be more widely used because of the new availability of isoxafludol tolerant traits on the market. So to leave you with some parting thoughts, zooming out from our specific research, what I hope you'll remember is that in the US and many other parts of the world, biotechnology is currently, and I expect in the future, an important part of modern agricultural practices. And so I think it's really important for those of us who want to enable sustainable agriculture with minimal environmental and health impacts that we understand these tools, understand their specific implications, and try to contribute to their safer and more sustainable use. In addition, something that my group is trying to think about is how we can move from our short-term research on these immediate environmental problems and try to enable some of these longer-term solutions. At the end of the day, we could develop the safest, most sustainable herbicide out there, and it may be only effective for a decade or so before these weeds evolve herbicide resistance. So if we really want to enable long-term sustainability, we have to work on not only these short-term problems, but also longer-term issues like trying to break down this herbicide resistance cycle that really limits the long-term use of our safest and most sustainable products. So with that, I want to acknowledge our funding sources, my wonderful research team, and my collaborators on the dicamba project. Really looking forward to the Q&A, and you can always contact me as well for further discussion after this event. Thank you so much.
Thank you so much, Professor Parker. That was incredible. The aspects of the long-term and short-term solutions, I think, based on the questions that are coming in, are definitely going to be part of the discussion. So thanks for bringing that up. Our next speaker today is Professor Anna Paltseva from Purdue University, appointed to the position of the Clinical Assistant Professor in both the Agronomy Department and the Horticulture and Landscape Architecture Department. Anna integrates community engagement and sustainable soil management practices to address the environmental challenges posed by urbanization. So take it away, Anna.
Thank you so much. Okay, here we are. Thank you so much for having me today, and thank you for those who join us. Today we'll be talking in the next 10 minutes on how we can build safer, more resilient croplands, starting from the ground up, literally. Our soils are under pressure, and of course it comes from like a legacy pollutants, industrial residues, increasing scrutiny around the food safety. And I will outline a path forward based on detection, remediation, incentives, and traceability. Let's start with the question we were invited to address today. What innovations and actions can preserve croplands as a safe, sustainable foundation for food systems? Soil pollution touches policy, public health, market access, and food justice, and it requires us to think holistically across disciplines and across sectors. Pollutants come from multiple sources. Heavy metals like lead, arsenic, cadmium come from mining activities, fertilizers, legacy pesticides. Persistent organic pollutants like DDT, atrazine, dioxins, they're used over decades. PFAS, the new forever chemicals that have been also around for decades, but we only started to study them in the last, 20 years. They're introduced via biosolids and industrial waste. Microplastics and additives from plastic mulch, compost, or even irrigation water also pose a big threat. We are seeing physical and biological changes in soils, acidification, microbial disruption, and declining nutrient cycling. This undermines productivity and increases the risk of contaminants entering the food and water systems. This is a fundamental issue, not only for agriculture, but also for the environmental health and long-term human health. We often think about big croplands in agriculture fields, but what I've been studying, and I would like to emphasize it in this presentation, that this pollution problems is not only for croplands, but also for urban spaces. And urban agriculture actually can provide us with food access, especially in underdeserved areas in cities. In many cities across the United States, urban soils carry legacy of industrial fuel, lead-based paint, vehicle emissions. Studies have shown, and something I study, is that elevated lead levels found often in low-income neighbors where interest in community and home gardening is often growing fastest. So in order for us to promote urban agriculture, we also need to ensure that urban soils are suitable for healthy produce. But there are solutions to have a cleaner soil and more like healthy food, is to use raised beds filled with verified clean soil and geotextile barriers, publicly funded soil testing programs, and education on safe gardening practices and produce choices. This is something I've been working with. Is to support these ideas, I developed the Urban Soil Guide. It's a field and lab manual designed for educators, practitioners, people, or anyone who work in urban spaces and want to understand what is the soil beneath their feet. It offers practical protocols for soil sampling, risk assessment, decision making, and helps communities to take ownership of their own soil health and make informed choices grounded in science. It's can be applicable not only this approaches in cornfield, but also in a community garden. To respond meaningfully to innovations and agriculture challenges, we first need to detect contamination, pollution reliably and affordably. Standard lab tests are quite accurate, and but rather they can be slow and costly and not always acceptable or not always accessible. So we need to come up with a new field deployable technologies that put data into farmers' hands. Portable X-ray fluorescent devices. I've been using this device for many years, and it offers rapid metal screening right in the field. Within 60-90 seconds, you can get a whole array of different heavy metals. Another example is biosensor kits. Using engineered microbes, fluorescence, in the presence of some toxins, it can actually reach 90% accuracy. Those are new innovations from Boston. Several US cities now offer subsidized soil testing to farmers and communities. There are also open access mapping tools, like in Washington, they have Dirt Alert that help flag high-risk zones. So making contamination visible empowers growers and planners to act early and wisely. This is also about transparency, because consumers increasingly want to know not just how food is grown, but also in what kind of soil. It brings us to remediation. So detection is the first step, and remediation is where the actual impact happens. The focus is not only on eradicating contamination, but on immobilizing toxins and restoring soil functions. So there are amendments and fertilizers that we can actually use. Phosphate fertilizers can bind lead and cadmium, for example, into insoluble forms. Lime and silicates reduce the solubility of metals in acidic soils. Biochar made from agricultural waste absorb metals, retain moisture, and also support microbial recovery. Compost overall improves organic matter and can reduce bioavailability of pesticides and metals. These strategies already been tested, and in my research we used phosphate amendments and compost in New Jersey farms, where we stabilized or immobilized lead and arsenic in farm soil, because the soil was contaminated with known lead arsenate pesticides back in 20th century. So it's really important to use new amendments, recycle organic waste that we can use to not only sequester carbon, but also improve soil health and immobilize existing pollutants. And also, it's really important, the verification part. So remediation must be measured through follow-up testing to ensure that actually the risk was reduced. Well, even the best technologies won't scale without infrastructure and policy support. We need systems that reward contamination reduction. The carbon market, for example, offers such a model. Farms with lower emissions receive credits or price premiums. So why couldn't we develop soil safety scores? Verified soil cleanup and traceability could translate into market value, compliance credits, or eligibility for grant programs. Maine, state of Maine, for example, is taking the lead by banning PFAS, the forever chemical, contaminated sludge, and funding farmers' relief and remediation trials. Public-private models, especially those that engage in community labs, input suppliers, and regulators are also essential. This brings us to traceability. So food traceability is evolving, and soil is becoming more and more pronounced in this story. Buyers are beginning to ask questions like, what contaminants are present in the growing environment? Were remediation practices in place? Can the source soil be verified as safe? So traceability tools that include soil health data will become increasingly valuable, not just for compliance, but also for branding, certification, and consumer trust. Soil transparency can protect farmers, reassure consumers, and reduce risk along the supply chain. So to close up, soil is an infrastructure. It stores carbon, holds water, feeds our food system. It supports every terrestrial ecosystem we depend on. Through better detection, practical remediation, economic incentives, integrated traceability, we can make soil safe as a standard, or soil safety as a standard. So let's work together towards croplands and urban spaces that nourish today and into the future. Thank you very much.
Wow, no, thank you so much, Anna, for that presentation and the overview. Absolutely gorgeous slides and overview to what soil is and how it comes into this conversation. Our final speaker today before the discussion is Professor Eric Mathis. Professor Mathis is Director of the Sustainable Food Economics Research Group and Professor of Agriculture and Resource Economics, the Department of Earth and Environmental Sciences at KU Leuven. Take it away, Eric. Thank you.
Thank you very much for this opportunity to actually dwell on what Anna also said in the end, which is to look at the economic incentives for making the application of these technologies happen. I will do so from a European perspective. In fact, I'm coordinating a European project called Soil Values, Enhancing Soil Health Through Values-Based Business Models, which is framed within the Mission Soil. The Mission Soil is one of the European Union also decided to have a couple of kind of moon shot missions, and one of them is actually focused on soil health, which means that the European Union is investing quite a lot in research to support research and development for soil health. And in this project specifically, we're looking at the incentives, the policy incentives, the public and private incentives of making these changes happen. And we're doing it by developing a framework, an evaluation framework, to help different actors evaluate the changes in soil health and especially also the economic elements of that. In different European countries, and we're also trying to figure out how you can combine actually different types of incentives to make it happen. Because if you look at what the situation is, the various speakers have talked about various types of technologies and practices that are available. or are in development, ranging from biotech, but of course from a European perspective, we are much more looking into what are called nature-based solutions, also carbon farming, but biofertilizers, biopesticides, biocontrol agents, but also to enhance soil biodiversity. So more extensive uses of solutions. But the problem is that the public funds, Anna was saying, well, we need public incentives, but these public funds are decreasing, and they're decreasing rapidly for all kinds of reasons. One of them is geopolitical reasons, where we have to divert funds for other reasons. But that means that we do have a large financing gap. So we have a 60%, for example, of European soils that are not in a good condition for different reasons. They may be contaminated. But actually, most of them have problems with either with soil erosions or with contamination just means too much nitrate in the water, for example. So it's not only heavy metals, but I suppose much more too high use of nutrients. So how to finance that? How can we actually leverage different incentives to make that happen? And basically, I want to introduce, Anna, you talked about ecosystems. I want to introduce the concept of ecosystem services. So all these practices, whether it's precision agriculture or carbon farming or increasing soil organic matter, basically constitutes a set of sustainable soil management practices. And these management practices, they produce some ecosystem services. And so these ecosystem services may be more carbon in the soil, or it may be less pollution. It may be a better water quality. And so the idea and our research is contributing to basically not only measuring these ecosystem services, because this is of course the tasks of the agronomist and the environmental scientist, but as economists we want to translate that in value. What is the euro or the dollar value of these ecosystem services? And once we do that, we can also investigate how to finance these practices or these ecosystem services produced. For example, in the case of soil remediation, you want to improve the soil quality or the soil health. And the question is, what is that worth to the different stakeholders? And can we blend different financial instruments to make that happen? Now, one of the problems of these ecosystem services, reduced pollution or higher biodiversity, is that they have private benefits. But if you only look at private benefits, we're looking at the farmer who may be willing to pay for the technology if it increases his profits. But many of these solutions, especially the nature-based solutions, may only have revenues in the long run, because you have to build up soil organic matter. But for example, using nature-based solutions to reduce nitrogen fertilizer probably won't happen in the first year of application, but maybe takes 5, 6, 7, 8 years to take place. And also one of the tension that we see, and that of course depends very much in the country and the context that you're operating is that often the land manager, the landowner is not the land manager, the farmer is not the land user. For example, in my country, in Belgium, 2/3 of the land is actually leased from landowners. Whereas, of course, many of these investments increase the value of the land and the benefits of which will go to a landowner, but maybe not to a land user. So there is that tension. So the long-term horizon, but also the land ownership versus land use, the property rights of it, that may provide problems. But actually, the biggest problem that relates to these ecosystem services is that they have public benefits, which is good, of course. But at the same time, it means that there is a free rider problem, because public benefits, public goods, means that everybody can enjoy the benefits of that. For example, erosion control, that's of course increasing crop yields for the farmer, but it also is beneficial for the whole community because of less runoff, less sedimentation in rivers and so on, just to give you one example, or less nitrate in the water has larger benefits. But the problem is that those that are the beneficiaries of these benefits is society at large, is, for example, a sector at large. And if you want to mobilize financial solutions, then, of course, that free rider problem arises. Why would I, for example, as a company, invest or help farmers in increasing soil health if my competitor also benefits from that and my competitor is not supporting that? So there's a coordination problem. And of course, one way of solving that coordination problem is using public funds. But as I said before, public funds are limited. So we need to combine public funds and find ways to leverage also private investment in this. And that's actually the biggest challenge that we are now doing research about. And basically, you have three sets of incentives that you can combine. One of them is technical support. Anna, you've been talking about basically verification, the fact that you have to measure and report and verify the changes. So that the development of this MRV technology is very, very important. In Europe, currently, we're trying to set standards for carbon in that respect. Because one of the problems of these verification technologies is they're often very costly. Lots of technologies are out there, but they're too costly for a farmer to adopt, just to verify, to measure the changes in soil quality. But technical support can be advice, can be money to develop projects and so on. The second area is related to risk reduction, to de-risk some of the technologies that are out there, because one of the reasons why, for example, banks are not willing to finance such new technologies or strategies is that they're too risky for them, or where, especially when you look at, for example, nature-based solutions. Take for example, crop rotation, and one of the nature-based solutions is to have more broader crop rotations, that you're not doing monocultures, but you're including, for example, legumes in your rotation of wheat, but maybe even 4 or 5 crops. But then, of course, there is an increased risk of finding a market for those crops, which means that, for example, off-takers may enter into contracts, or you can have insurance companies that can also be part of the equation in de-risking your project. And then the final point, of course, is, as I said, if the rate of return of your technology is not high enough to benefit just the farmer for all kinds of reasons, but also benefits society, additional financial support is necessary. And I gave the example of price premiums or carbon credits in the offset market, for example. These are examples that are already existing, but there are many instruments that can be used to increase the rate of return of such projects that introduce new technologies. Basically, there are 4 mechanisms to increase returns, and one is to use cost sharing, so to change your practice, to change ecosystem services. One way is that you subsidize your farmers, for example, by subsidizing part of their costs. A second mechanism is that you use an insetting approach, which means that buyers are offering, for example, a 10% price premium. For example, the beer industry is doing this for paying a 10% premium for barley for beer brewing. For farmers that invest in healthier soils. That's called an insetting approach, where you have a link between a buyer and a farmer. The problem is when you're talking about rotations, of course, if you would do that through carbon farming, is that, of course, the next year there will be another crop on the field, and then, of course, the coordination problem between different types of buyers arises. And then the third mechanism is the payment for ecosystem services, where you kind of remunerate the farmer for the ecosystem services that are being offered. And again, for carbon, that's being established, but for other ecosystem services, like nature credits, for example, this is still in development. And then the fourth mechanism would be, well, the fact that your land becomes more valuable by remediating it, for example, means that your asset increases in value, and that is also a mechanism that you could use to attract investors. So how to get these investors into the picture? Well, the problem is investing in nature, for example, does not provide a lot of return. The rate of return of investing in nature is very low, which means that you need to combine different sources called blending finance, or you need to get public or philanthropy into the picture. But that in itself isn't sufficient. You also need to raise the rate of return, and you can do that basically in two ways. Either to make the investment mandatory, so going from a voluntary market situation to a mandatory market situation, like we did in the European Union with the ETS, the carbon market, is basically a market that works because it is mandatory for the industrial sector to reduce carbon. There is a tax on carbon, which means that there is a market for credits. There is also an offset market to do that. Or the second option, which is an option in development that the finance sector is also very much interested in, is that you turn the improvement in your land, to the improvement of ecosystem services, into an asset, an intangible asset that can then be traded, can be used, for example, to get a loan from a bank. So that's kind of a new financial product that are also in development. And so our research challenges that are addressing some of these issues is, one is to link these practices to the provision of these ecosystem services, because there are some synergies, there are also some trade-offs between ecosystem services, between production. Secondly, to develop standardized data and methods for measuring and valuing these ecosystem services, which is particularly a problem for biodiversity. And thirdly, to also to derive principles for blending these different types of incentives, because of course we don't want to over-subsidize, we don't want to subsidize industry. So governments are also always very reluctant to do those things, because we want to make solutions work, but of course not over-subsidize the private sector. And then lastly, to map preferences of land users and stakeholders to these incentive schemes, fostering sustainable soil management practices. Thank you. Looking forward to the discussion.
Thank you so much, Eric, for sharing a really nice kind of just summary of kind of everything that we talked about and how it all links together from an economic perspective. I would ask now we only have about 15 minutes for discussion, and we had a ton of really great questions in the chat. So I'm the one that's going to be kind of housing this discussion part of the day. We probably will only get to 2 or 3 questions probably, but I hope that some have been answered, I think, by some speakers, and we might be able to figure out a way to present answers to the questions after the presentation as well on LinkedIn. So I wanted to kick us off with a comment that was a question in the chat, and it kind of relates really well to Eric's last presentation, which is about the uptake of all the technology that we talked about today. There's a ton of research in the field, and especially now in the last 10 years or so. And for a farmer, potentially, it may not be clear exactly what technology they should adopt and when. And there's many things on the market that can all claim similar things. It might just be almost luck on who's there and who's marketing what at a time. So where does the burden lie in this communication? Is it with the regulatory bodies? Is it with the salespeople? Is it with the scientists? Is it something else entirely? Yeah. Eric, yes.
Can I pitch in? I think one of the I mean, in Europe, the answer is the advisory services that we still have, and in an American context, that would be the university experimental stations that take a neutral stance in this, because the risk is of course that the private sector of course wants to sell their solutions, which is not a biased which is by definition a biased perspective. But a farmer should be able to have an unbiased view on that. And so strengthening the advisory services, however that is organized, because it's organized very differently in different countries, I would say is a key in solving this issue.
Well, I would second it. It's actually something that Purdue does, I think, quite well. As a land-grant university, the university mission is exactly what Eric said, is to share this education and innovations and with the farmers, because it's a part of the university mission. And through a lot of experimental station and extensions, they actually have a lot of events. And since I'm here for a little more than a year, so it's like a new eyes to see the system. And I think if we could expand this nationally and globally, of course it comes back to resources, but I think this is a model that works, and there's a lot of partnership with the farmers, also because they're former alumni. I think this is very interesting. As a new faculty, we have tours, and they give us tours like every August, like for the first 3 years, and they take us around farms where we learn what farmers are doing. And there's throughout the year, they have events when farmers come to campus, or there are different events. They can always call up. But I found it very interesting, this connection with the alumni who are actually farmers in industry. And this is how the model is built on this relationship with the farmers and industry, but they are university former students.
Yeah, maybe I could jump into. I think it also depends on where you are. For example, I'm in New England, northeastern United States. The average farm in Connecticut and through much of New England is 40 acres. So, and just as Eric said, I work in an agricultural experiment station. Our mandate is to work directly with farmers. However, if you want to talk to soybean farmers in Minnesota or Iowa, probably addressing commodity groups is the first step to take. Certainly talking to regulators is a part of this, but I do think talking to the major agrochemical companies is as well, because they definitely have interest there. If you want to go to the big farming operations throughout the United States, I mean, you're a lot of those 20, 30, 40,000 acre farms, they work a lot with seed companies and, your Bayer Crop Sciences and your Corteva and all those. So, partnering with companies, eyes being open is definitely a strategy as well. So I think you just kind of got to see what the landscape is and take the approach that works best where you are.
Yeah, definitely interesting. Maybe on the idea of where you are, I think there was a lot of questions in the chat, and Jason did get to answer a bunch of them about developing countries and kind of how the landscape is totally maybe different. And we talked a lot about in the beginning, light at the end of the tunnel and a train, and the train can be moving different speeds depending on what context and scale you're working at or where you are in the world. I wonder if if the rest of the panelists wanted potentially a chance to comment on some of those questions. There were quite a few about developing countries, and these advanced technologies are also expensive sometimes, but they can do a lot of good. So if anyone wants to comment on that too.
I guess I can say on the biotech side, so this is a little bit outside of my scope of work. Like I said, we're really focused on regional issues here in the Midwestern part of the United States. But I do regularly attend conferences and talk with folks working on the biotech side. And there is a ton of products, both in the research stage and the commercialization stage, for biotechnology to address these challenges in developing countries. And so I think there's going to be more and more of that in the very, very near future. And so I think it's definitely something to keep an eye on if you're interested.
If I may also add on that, one of the things that we've been working on quite a lot is the idea that as we make our data more available, so there's the concept of fair, findable, accessible, interoperable, and reusable. And as we build these machine learning models, if we can make those open access as well, and really, sort of democratize the availability of those models and the data. So one of the things we've been doing a lot with collaborators is developing not just the really technical model, but also the web interfaces that allow people who don't have a coding background and you don't need then to be able to do modeling, to be able to work with and play with the data and visualize things. So I think that idea of really democratizing both our data sets, our models, and the information that we can share, so that it can then be tailored. So, I mean, if we do our studies in a European context or a North American context, can we then use models to say, well, okay, what about the soil in Brazil, or what about the soil in India, or what about the climate conditions? So how can we then, adjust based on the local conditions? Or, one of the things that I think about a lot is, we often assume, and I think, I'm not sure if it was Anna that talked about it, the history of the soil. So the urban soils tend to have maybe metal pollution, but some of the rural soils will have a whole history of stuff that's been added that maybe you don't know. And, one of the potential concerns with nanomaterials, or even with biochars or anything like that, is that they may then concentrate up or absorb those things that were in the soil previously. So, we need to think about some of these broader issues as well. And what is the history of the soil? What has been put there before? And, once we think about it, then we can say, oh, okay, well, if this is a case, then we can design materials that also then, promote the biodegradation of those. So, everything is an opportunity as well, as long as we know what the answers are. But from my experience, I think India is probably one of the countries that's moving fastest in terms of nano-enabled pesticides. There's quite a number on the market, and there have been a few market reports recently about the potential, and how how different areas under different regulatory environments are moving at different paces.
Yeah, thank you. Thank you so much for that overview. And I think you tackled a bunch of other questions in the chat with your answers there. So that actually makes my job a lot easier. So thank you, Isolt. I did want to maybe have one final question that everyone could potentially answer. And this one comes inspired by Jason's slides, actually, where it was about, the 10 years ago, this field looked very different. And it's almost just a thought question about where do you think in another 10 years, what do you think a success would look like? What do you think potentially failure? Whatever you want to answer, but 10 years from now, what do you see as this field coming together to answer some of these, this train coming? How is this field going to work together to accomplish anything? I don't know who wants to start, but blue skies picture.
I can start off. I see there's quite a bit of fintech startups combining, as was said, artificial intelligence with, for example, also remote sensing, which is a very strong tool. But any kind of data. I mean, and of course, in data-rich areas like the US and Europe, that may work well. But I think these tech fins, they're really leading the way in some of these areas. And I think the challenge for the global south is where you have a data poorer environment, especially below ground, is to kind of find solutions for those situations. Because honestly, the delta is much bigger in the global south than it is in the global north. So as an investor, I would be willing to invest much more in the global south than in the global north, because as somebody in this chat also said, governance is a really big issue. And this is, if we can get governance figured out, for example, in Africa, that would really make a step change.
Well, I can jump in. Maybe I'd start off by saying I'm actually, in spite of how dire the picture seems to look, I'm actually really optimistic, because I think, and today was a perfect example of this, we have the tools to address this problem. Now the problem is a moving target, right? It is a train coming at us, and I think the thing we need to recognize is that the solutions, it's never going to be one solution, and every system that you're in, whether it's sub-Saharan Africa, rural India, or Connecticut or California, the solution is going to be different. So I think what we need to recognize is that we're going to need a very large, robust toolbox that's going to include biotechnology. It's going to include traditional plant breeding. It's going to include nanotechnology. And what we need to do is, look at the system where you're trying to fix the problem. And then once you have that toolbox there, then you can pick and choose. So our job as scientists is to fill the toolbox. And 10 years from now, I think it's going to be full because it has to be full. I mean, we have no other option at this point. I mean, food insecurity is on the rise everywhere. There's not a corner of the planet that we're not seeing that. And the challenges are getting, the challenges that are making that worse are not going away. So I think we're on the right road, and we'll be there sooner than we think.
Just picking up on that, one of the things I think will be the future as well is this idea of vertical agriculture. So right now we kind of, we have a field and it has one layer of whatever the crop is. But a lot of work now is going into sort of the hydroponic systems where you can stack them. So in the same way that we build our urban environments higher density, I think there'll also be really big opportunities around vertical agriculture, and those are more closed loop systems. So again, you have savings in terms of water. You have the ability then to regulate that water, to have more precision over when you need to fertilize or when you need to enrich. So to me, that's I think one of the really exciting areas as well.
I'll just add that I think it's really important that we try to, think about where farmers are at, in their specific context, and try to make sure our work and efforts are responsive to that. I think ideally for me, as someone who cares deeply about the environment, but also agriculture, the more I can find win-win scenarios that both protect the environment and help farmers, I think the better. For the example of drift, no one wants the herbicide to move into the atmosphere. The farmers want it on their field. We don't want it off the field. And so if we can develop better tools to address that, that can be these win-win scenarios that I'm really excited about.
Well, and I hope from my perspective on pollution, they will find new ways and easier ways to map it, and on a positive note, new ways to remediate it. And I think microbes and nanotechnology and like fungi, they will help us to do it. So hope to see more and more innovations on that front in the near future.
Awesome. Yeah, thank you. Thank you so much. Thank you for our speakers. I know that this is virtual, so you can't hear applause, but I'm sure that the people that are listening and tuning in are definitely clapping for you right now. Thank you so much. And thank you so much for our listeners. Thank you for being part of today's webinar, and people who are watching the video recording, thank you for being part of it, and please continue the discussion. On behalf of myself and the newest journal, Chem Circularity, Richard from Cell Report Sustainability, and Shan from One Earth, we welcome you to continue on the conversation by checking out our journal websites, keeping connected with us and all of our speakers who generally just welcome further communication with them as well. Thank you so much for being here today, and have a very lovely rest of your day, night, morning, whatever you have going on. Thanks.
Thank you so much for having us today.
Bye, everyone.
Thanks all. Bye.